Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing you not one, but two really awesome guests involved in creating a better tomorrow for many people out there. Today, we honor being joined first by Michael Burton, who is the Chief Executive Officer of the Diabetes Research Institute Foundation, a philanthropic organization that funds the Diabetes Research Institute, which is one of the largest and most comprehensive research centers dedicated to curing diabetes. Michael has more than 3 decades of experience in leading these high-impact philanthropic programs, cultivating strategic relationships to secure such transformative funding. Prior to this role, he was advancing the missions of some of the nation's most dynamic and trusted institutions, including Princeton University. He was at the Pew Charitable Trusts, is also at the American Association for Cancer Research. He also, most recently, prior to this position, served as president and CEO of Gateway for Cancer Research, a Chicago-based nonprofit engaged in the early funding of clinical research. Began his tenure there as chief development officer. Also spent time here in the Philly area at Fox Chase Cancer Center as their senior vice president, chief development officer. We're also joined today by Professor Dr. Matthias von Herrath. He is the scientific director of the Diabetes Research Institute, as well as the Stacy Joint Goodman Chair at the University of Miami School of Medicine. They're leading DRI's mission to accelerate research to ultimately delay, prevent, and ultimately cure type 1 diabetes. He is widely recognized for his work at sort of the intersection of autoimmune disease, endocrinology, and sort of this connection potentially with virology and understanding how cells are ultimately attacked by the immune system and destroyed. He served as professor and director for the Type 1 Diabetes Research Center in La Jolla Institute for Allergy and Immunology. Also currently serves as VP and Senior Medical Officer at Novo Nordisk, and he earned his medical degree from Fieber Medical School, also completed a PhD in biochemistry. A lot to get into today, a lot of really cool topics. Honored to have them both. Michael Burton, Dr. Matthias von Herrath, thank you so much for taking time to come on the show.
Thank you.
Super happy to be here.
Yeah, no, it's great having both of you. I'm looking forward to delving into a bunch of topics. Would like to start off by handing things back to you guys for a little bit, just to talk a little bit more about yourselves. Michael, if you could sort of give us a little of your background, sort of what got you into the sort of the philanthropic space. I know you did spend a lot of time around the corner here at Penn, at Jefferson, Temple, Fox Chase. Give us a little more of your background story.
Happy to, and thank you again for inviting us on. So I have spent a big part of my career in higher education, working for universities, helping universities raise the resources they need to do the extraordinary work that they do. I was at Princeton University, I was at Temple University's Fox School of Business, and also the University of Pennsylvania, where I went to school and served primarily in development roles, but also staffing the then president, Judith Rodin, the first female president of an Ivy League institution. Extraordinary experience. I was very happy in higher education, but received a call from a recruiter about a position at the American Association for Cancer Research in Philadelphia, a very high impact, very important organization in the fight against cancer. At that point in my personal life, unfortunately, I had a lot of loved ones who were grappling with this disease. My father, my mother-in-law, my sister. Sadly, we lost all three in quick succession. And I thought maybe that was the universe trying to tell me something in terms of this position at the AACR. So I took the leap. And for 15 years, I was working in the field of cancer research and was working in the field at a time, I think, that was really extraordinary. And I had the privilege of seeing some of the most important advances, I think, that have ever taken place in the field of cancer research and treatment in those 15 years, notably the area of immunotherapies. And so, again, what a privilege that was to see that tremendous progress. I received another call from a recruiter who talked to me about this position. And I was a little hesitant at first to move on from oncology because I had invested so much, had the privilege of working with some of the most important players in that space over the years. But, you know, the more I looked, the more I spoke with the players, and certainly the more I spoke with Matthias, the more I realized this made perfect sense for me professionally. I feel like the field of diabetes is very much where the field of oncology was a little while back, and that is poised at the precipice for extraordinary discovery, and then the translation of that discovery into new, innovative, life-changing therapy. So it is a pleasure to be in this role. I've been in this job for about a year and some change, so I'm still very much new to the organization. My head is still somewhat under the hood, still kind of learning the ropes, but again, an extraordinary opportunity. I'm very happy to be the Chief Executive Officer of the DRIF.
Excellent, excellent. And Matthias, I mean, you, as I mentioned in your bio, you know, actually going back for a couple of decades now, you know, you've been at this very interesting intersection in your own research in terms of endocrinology, autoimmunity, and sort of this virology connection, an area that's extremely hot still in 2024. You wrote this really cool review back in 2016 on sort of where we were with the viral paradigm. Would love to hear a little bit about your background, what got you focused on this, and, you know, a little bit about it, as you were saying in this paper. What are the suspects nowadays? And we'll be getting more into the programs, obviously, to talk about this, but it'd be interesting to hear sort of your thoughts as where we are in 2024 with this concept.
Yeah, I'm going to first take a little bit the ball from Michael that he threw to us. Everything right now for us coalesces in our paths at the Diabetes Research Institute. It's a really very special place in terms of collaboration and trying to bring things over the finishing line for type 1 diabetes and for the patients with type 1 diabetes. In terms of the background, that's a long story. I got involved in medicine because I lost my father very early in life, and it created a substantial insecurity in myself dealing with health issues. It was sort of an event. I was only 7, 8 years old. And it was a bit like, how can this happen? And my father didn't die from diabetes. He died from a brain aneurysm. But it instilled in me this drive that I want to make a difference. And I first worked in medicine, in oncology, actually. So we have many intersections there. And then I went for research now a long time ago in the US in 91. And indeed, there our main interest quite immediately became this intersection of viruses in diabetes. And I will revert back to that because that's a little bit of a conversation. And as I progressed, we used to, like many of us, work in animal models initially in the 90s. And then in later in my career, I joined Novo Nordisk because I felt I wanted to translate things to patients with big trials, and a large pharma company like Novo Nordisk seemed to be the right thing. And we have actually made a combination therapy trial and just recently reported an antigenic tolerance trial. We have made good strides. Yes, the viruses and diabetes, you know, what is so fascinating about the immune system that we are studying is that sometimes the immune system goes awry, like in diabetes, and attacks a single cell. But on the other hand, sometimes it also goes awry in cancer, and actually should attack a single cell to eliminate your metastasis, and it sort of fails to do this. And this fascination with this, on the one hand, the specificity of the immune system, but on the other hand, how it learns to attack specifically and when not, that has fascinated me for a long time. And of course, we don't understand all the rules, but we understand better rule sets. And certainly viruses are a good paradigm in this respect, because the immune system is made to fend off pathogens, and it usually attacks virus-infected cells. And it also should control chronic viruses, sort of like it does with cancer. So we have all herpes viruses. The immune system controls these. We are stuck with the herpes viruses, but the immune system has this exquisite surveillance crew. But sometimes the immune system goes wrong. And the question there is, how does this work? And it seems in type 1 diabetes, it's not so much a problem of a systemic mal-regulation of immunity. It's a local problem. There's something locally in the pancreas that happens where the immune system all of a sudden sees beta cells that it destroys and attacks. So it's like you pull we always say we pull this Harry Potter cloak off the beta cells, and the immune system goes like, whoa, maybe I forgot something there. Now I got a strike here. And that's where viruses become important. We were very hopeful in the beginning that we would find one single culprit. How nice would have that been? You could tell patients, here's the virus, and that causes type 1 diabetes. And now we're making a vaccine, and we're all going to be cool. We don't need to worry about this disease anymore. Unfortunately, like often in science, things throw a curveball, the real biology, really. And we had to learn, if you want to ask where this story stands now, that indeed virus infections can probably play a role in sort of decamouflaging beta cells to immune system, but it's more a general phenomenon. So it becomes more a multiple hit situation, and it's not specific for one virus. It's more how many times you're unlucky to hit your pancreas. If you're already prone to develop diabetes, you get hit X amount of times, and that's going to set you forth on a bad pathway. So it's more complex, unfortunately, also than treatment-wise. And it's maybe comparable in a way with climate change, that you have these multiple hits that can occur in various orders and from various directions. And you're pushing a system that is initially very stable to a brink where it cannot stabilize the sugar levels anymore in the blood. So that would be my answer to the specifics of your virus question. But we can talk about this for a long time. It's very interesting altogether.
I very much appreciate that. And the reason I wanted to bring it up, not just to show sort of what you've been doing, but when we get into some of the discussion about the current programs and then the principle sort of how one doses a combination in these particular cases, when you're looking at a cure and figuring out there's not going to be a single magic bullet here, but you got to put stuff together. We're going to come back to it, but I appreciate you, Matthias, giving us that part of the story. It's an important piece here. Before we get into the organization, I mean, I just want to sort of do a current sort of state of the T1D landscape of where we are now, in the sense that here we are in 2024. I think current numbers, we have somewhere over 9 million people worldwide diagnosed, about a little over 2 million of them in the United States. The projections are, off the scale where what's going to be happening 2040, 2050. Clearly a significant unmet medical need. I would like you both to take a little time, and you can ping pong back and forth, sort of talking about these numbers, but at the same time, would love to hear a little bit about sort of the respective challenges in both your offices. Michael, you from the perspective, obviously, when you're out there talking a T1D story, clearly you run into people that are interested in melanoma and frontal lobe dementia and one of 10,000 rare diseases that fundraising competes with. So I'd love to hear a little bit about the types of things that you run into when you're on the road. And Matthias, at the same time, clearly the DRI mission is a cure. But at the same time, when we have other parts of this diabetes equation out there, whether it's insulin pumps or artificial pancreases or whatever the case may be, again, talk about the respective challenges in your shops, let's say, within the organization.
I think we have a very good division of labor in a way between Matthias and me. So the foundation, which was established 52 years ago by 5 families that were, deeply affected by this disease. The foundation is focused solely on raising awareness and resources funding so that Matthias and his colleagues, the very talented, very passionate clinician scientists and researchers at the DRI, for them to do the work that they do so well. And that is our job. Our focus is at the foundation is on raising that awareness, raising that those resources, all with the goal of curing this disease. That is our focus. That is our mission. And we are on a path, I believe, to the cure. Along that path, we are, and Matthias can speak to this much better than I can as the scientific director. But along that path, we have made discoveries. We have advanced the field in ways that have made a tremendous difference in the lives of people who are navigating this very challenging disease, this relentless disease. So on the path to that cure, we've discovered the impact of and the value of insulin pumps, of CGMs, of better insulins. The Institute itself is focused and has pioneered the field of transplantation, beta cell transplantation.
Yep.
And so, these are short of the cure, clearly, but they have these discoveries have had a tremendous impact on this disease and on the lives of those who are managing this disease. But again, the division of labor is clear. The foundation focused like a laser on raising awareness, raising resources from individuals who are philanthropically inclined and have significant philanthropic capacity, raising resources from foundations that exist to support this important work, on corporations that are focused on the diabetes space as well. Essentially, any individual or entity that wants to join us in the fight to defeat this disease, to cure this disease, we seek and encourage that partnership.
Matthias?
Yeah, I mean, we indeed work well together. We have lots of synergies, and we have also lots of fun together, which I enjoy a lot. As Michael alluded to, we celebrate all the small victories. The cure for a disease is not a binary thing. Well, sometimes it is, but very rarely it is. You have to sort of celebrate all the small victories. It's like learning to fly an airplane. There's lots of little milestones that we should all celebrate. Among them, of course, you alluded to it, improving insulins and having better metrics for continuous glucose monitors, closed loops, pumps. This is important for patients. There's absolutely no doubt. And these are victories that need to be celebrated. They improve HbA1c for all the patients, type 1 and where applicable for type 2. So these are important building blocks in the patient's eyes and our eyes. This is, of course, not the final cure. There's more things to be done. We need to put stem cell derived islets into people. And the barriers are, we would like to do this without the burden of immunosuppression. There's exciting new advances in that area. We can get into it if you want. We need to amplify beta cells, adult beta cells, that you grow more of them. There's also exciting new advances in this area and all areas where the DRI is involved in. And we need new devices where you have enough oxygen coming to the beta cells, but at the same time provide them with enough protection so they're not immediately destroyed. So these are all very good advances. You asked, though, about the overall landscape. Of course, that's not particularly easy. It's a reason why a place like the DRI is very important, because of all the good work the foundation does. Right. And here's two things that I think are quite important. One is, of course, we compete also market-wise in large pharma. You see this with type 2. The type 2 market is much bigger. And so type 1 is always a little bit the sickest child compared to that. I personally feel it should be. It is absolutely fundamentally important. You can hear my passion here, and we got to fight for this. This is very important, right? So there is a competition. There is actually a project that might make it easier. The DRI plays a role in this. We have in the US, out of our type 2 patients here, probably somewhere between 3 and 8% are misdiagnosed with type 2 and actually have type 1 with multiple autoantibodies. That makes, because we have lots of type 2 diabetes, that makes the type 1 population potentially up to 3 million people bigger as adults who are walking around with type 1. So it might not be as much of a sickless child as we think. It might actually be bigger. The other place, and I think people in general have to be aware of this. It's not anybody's bad intent, but the funding landscape in general, how it has evolved in academic research, is at a very complicated hiatus. And again, I'm not pointing fingers. It's mostly NIH that funds investigators, but the funding is very difficult to get at this point. The grant approval line hangs around 9 to 11% on one round. Of course, then cumulatively it gets better, but that's very low. It used to be, when the system of peer review was created, it was much higher. That puts extremely strong demands on investigators on timelines, and because of that, having foundation funding to do new creative ideas is so important. Because out of this strange system, what comes out of it is often that things get funded where the result is already almost clear that it's going to come out in a certain way, because it has been in waiting also for so long. Right. So we lose in a way productivity, which is not good, and it takes too long. And one thing that signifies this is that the age of a normal investigator to get their first R1 funding from NIH is now well high in the 40s. But you compete with that on the other hand, people say, and that's bad for us because we are older, but the most important discoveries are made by young people, early in their career. We have to keep this in mind. We got to foster these people. We got to keep them there. We want the young, bright minds. A big task also of the DRIF to keep these young, bright minds in a research enterprise so they can perform. They have the bandwidth, the runway to perform at their best. So that would be my short take on this, and why the DRI is a special place, why it matters in addition to the general ways of funding research, to have additional ways to support sort of the creative outbursts in real time, to then solidify new programs and push the boundaries, and with that, the little victories faster, right? That's very important. It's more important now than when I started, and more important when these systems were created, because it's just really, the air is thin if you're a principal investigator. It's not easy.
Right. No, I'm glad you brought all that up, especially the grant funding and the, I've had some of your friends on the show from Novo. We've talked GLP-1, and it's going to be interesting. We'll talk a little bit about some of the interesting mechanistic overlaps that you've been publishing on, in terms of how GLPs drugs for type 2 could potentially be helping out in sort of the combinatorial ways you approach sort of curing things. Before we get into the individual programs, Michael, I was just wondering if you could say, because we've been talking about the foundation, and Matthias was talking about the institute. Talk about the DRI per the Center of Excellence at the University of Miami, and because I just want to put this perspective for the audience, because this is not a small collaboration. This is a major university medical center. It's about 4 or 5 years ago, I had Camillo Ricordi on the show talking about the work in islet transplantation. Say a little bit, because I think that relationship goes back to, I guess, the inception back in the early 70s, correct me if I'm wrong there, but a little bit of that story as well, I think, is important to understand that, and there's a $50 million gift that just went to them in 2023. So pretty decent sized operation there.
It is. And I think the arrangement, the relationship between the Institute, the foundation that I have the privilege of leading, and the University of Miami is unique. And it is incredibly productive. Over the years, the foundation has given in excess of $300 million to UM and to the Institute to advance this important work. And, this, I guess it bears restating that the foundation is a separate 501c3. We're independent of the Institute and of the University of Miami, but again, we exist to support this important work. And we've been at it for some time, as I said, over 50 years. I think the donor community right now, broadly defined, is very enthusiastic about the opportunity to invest philanthropically in this work. They see what I was laying out at the beginning of this discussion, which is the tremendous potential right now in this field. And I think the people who support the mission at the Institute and at the foundation are people who have been, by and large, have been affected by this disease personally. They know and have seen up close and personal how devastating this disease can be, and they want to do something about it. And that cohort of donors really represents, I think, the lion's share of our supporters. But there's this other cohort of donors to the organization that I think is really interesting, that their personal experience with the disease is maybe more tangential. But what they see is an opportunity to have an impact in a disease that is, again, devastating and affects so many. So they're in the game, so to speak, to solve this seemingly intractable challenge. And that's why they're supporting us, because they want to see this progress. They want to see one day a cure. So we engage all sorts of people around the mission of the Institute. They bring different motivations, but they all want to see us defeat this disease once and for all.
Matthias, time to get into the research progress. Michael, you could rest for a little bit because we're gonna. Well, I really want to spend a little time here to sort of look at the breadth of what's going on, because again, sort of thinking back to this cure paradigm, we have the need to suppress the immune response. We have the need to sort of regulate the immune response properly, and then ultimately the big R word, regeneration, and whether we're talking about islet transplants or stem cells or any other unique regenerative biologic therapy that may be on the horizon, a lot comes together and ultimately creating that cure. Talk a little bit about sort of the research program layout, how you go about sort of finding the best primary investigators, evaluating the best drugs versus biologics versus cell therapies, and take a little time with this one because I really want the audience to appreciate the scale and the scope of what's going on here.
Very happy to, and I'm going to take the ball again from Michael that you threw me. I think what you're mentioning is center also for our research program, and if you want to summarize it in one word, is a collaborative spirit, and it's pervasive at the University of Miami. At the Institute attracted me to there, and it also the type of people we want to see, because the science dictates this where it is at this point. We want collaborative people. We have probably in general driven the scientific enterprise a little bit on the overdrive of competition. There's too many elbows in my opinion. And, all systems need to be adjusted from time to time. Of course, you don't want a completely uncompetitive system, but you also don't want a system where everybody has their elbows out all the time. And I think University of Miami, DRI, this is a sweet spot. And we initially discussed what is so important in terms of the big medical departments, but also the cancer department that's very big at the university. Sure. This is the yin and yang. We need to understand why cells sometimes attack cancers or don't attack cancers, and we will learn vice versa how this works in autoimmunity. Now, you asked me, you want, of course, the concrete programs, and we can spend time on that. I think there's a very fundamental realization that relates to islet transplantation as well as the natural history of the human type 1 diabetes that we actually studied for over 15 years in human pancreas samples from organ donors. Mm-hmm. And that has really opened our eyes how this disease evolves. It's different from animal models, and we have to appreciate at this point the differences. What happens in the human disease early on is that the beta cells start putting molecules on their surface, notably MHC class 1, and that in conjunction with some processing defects in insulin, so that you present to the immune system all of a sudden molecules with pieces of insulin that usually aren't there. That makes the immune system really nervous when there are new things showing up, because the immune system thinks, maybe it thinks it's a virus. The immune system is geared as a system to recognize things that are new that shouldn't be there. So anything you change that wasn't there before is always risky with our immune system, because it might confuse the immune system. I think it's as simple as that. But what that means therapeutically is there are intrinsic defects in the beta cell that could be either epigenetically or genetically. It could be innervation of the pancreas. There's some evidence for that. That aggravated maybe by some repeated virus infection. All of a sudden, the beta cells become visible to the immune system, and they, in a way, contribute to their own demise. This is very important to understand, because it's also relevant post-islet transplant, because you need to keep these newly transplanted babies, so to speak, hidden from the immune system. That's how that has to work. That means therapeutically, you all of a sudden have two arms, and that has been underappreciated so far. One is you need to keep the immune system back. That's the conventional approach. The second is you need to see that the beta cells becoming recamouflaged, and we can at least discuss in a few minutes how GLP-1s and maybe other drugs might fit into that space. And I should be very clear, we need trials on that. I'm not recommending any, definitely not any off-label use. We need to understand this, but there's exciting science that we can certainly share on that. And on the other hand, we need to tune the immune system. I think what is critical for the immune system, we cannot misunderstand that diabetes is a systemic immune defect. Patients with type 1 diabetes might have comorbidities, but they don't run around with lupus and arthritis and this and that with a systemically dysregulated immune system that are catastrophic, like an IPEX syndrome where you have no regulatory cells. This is a very specific problem to the pancreas. Like alopecia is a specific problem, like vitiligo is a specific problem. And we have to appreciate that this and a specific problem from the immune system needs to be ultimately treated specifically. And when we bring these two together, we also will have safe enough drugs. A drug that influences locally the beta cell to recamouflage it, and a drug for the immune system that ideally teaches the immune system to not recognize the beta cells again in a peacekeeping way. It's sort of like a deployment of selective peacekeeping group, but not saying generally, oh, we shouldn't have any military, the immune system is bad. We evidently all going to need it. We're all going to get infections, things like COVID and other things. We're going to need our immune system. We also need it to combat cancers, endogenous viruses. This needs to occur specifically. Otherwise, the patient gets sick. And there's good advances. It was, I think, just press release today that we had an approach of antigenic tolerance that has just completed phase 1, which is good. And we have published before, if you want to come into specifics, that GLP-1 drugs can be used to recamouflage the beta cells. Maybe there's at least evidence that patients who, after recent onset diabetes, were put on these have an improvement of HbA1c as well as preserving beta cell function, as evidenced by C-peptide. So that's not bad. There's also a published paper from us for in vitro organoids, mini beta cell organoids, that when they are under immune attack and you add a GLP-1, they are less sensitive to lose their function through this immune attack. So this evidence is good. What needs to be done, as I alluded to in the beginning, we need a randomized control trial, ideally in stage 1 type 1 diabetes, testing these drugs and see whether they can help with the prevention. Not alone, ideally, in conjunction with an equally tolerable approach that re-educates things. And conversely, if you're talking about the cure, we alluded to it in the beginning. In a cure, the key is that we don't have to immunosuppress patients lifelong. You can probably say if you get an islet transplant or a stem cell-derived islet transplant, look, you need immunosuppression for a limited amount of time to get this graft over the hump. I personally think that will be tolerable and a risk gamble that patients can take. Where usually the problem arises with systemic immune suppression, if you take this for decades, your risk of getting cancers, re-emergent herpes viruses, and having immune problems becomes higher. And so if you take it short, that would be okay, sort of like in induction therapy. Yeah. But then you have the problem, what happens afterwards? And this is where a lot of research also at the DRI comes together. We need these maintenance therapies that have the same classing in terms of low side effects that we could use also in early prevention of the disease. Mm-hmm. But where really the DRI comes in and research is needed, we need to find a sweet spot here because the situation will be much more complex. We will have a device that holds the newly generated islets or beta cells, at least in the holding tank. They will have contact with blood vessels, so the immune system can reach them. They will be happy with the oxygen they get from the blood vessels. They will not be very happy revisiting the immune system at the same time. So we need to deal with this immune system. And that can be done in two ways. One is that you have local immune suppression in the device that gives you pharmacologically an upside because you act more locally and less systemically. The other thing that needs to be happening is that you ideally genetically modify the stem cell derived new islets so that they are not expressing these decamouflaging recognition molecules. And in my mind, this will not be binary. This is not black and white. There will not be an islet that will never get recognized. They will, as much as there will not be a device that occludes the islets and there's always enough oxygen, as much as there will not be initially a magic immunotherapy that you take one pill that resets your immune system and just the beta cells don't get attacked. This is like wishful thinking, maybe 100 years from now. What we can grasp now, what we need to define in a collaboration with academic settings like the DRI, and then the companies and ultimately large industry, if there's sweet spot. We need this sweet spot. Somewhat camouflaged islets, a good holding device that is good enough to locally supply the islets, and with that, a reduced immunotherapy with induction maintenance therapy. That will get the door open. And once the door is open, meaning patients go like, whoa, this is maybe a therapy I could take some risks, but they're very limited, and it might get me insulin dependent, at least for a while. That will open the door, just like it happened in oncology. Michael knows this probably much better than I do. In the early days when I practiced, every leukemia was a death sentence. This has been entirely rewritten to refining the therapies and the combination therapies, right? We were celebrating the first patient, I remember this was in Freiburg University Hospital, who had survived the leukemia. It was like far beyond. Everybody else was dying. It was terrible, right? And look at where we have come now with these combination therapies in oncology. And this, once the door is open, this field will run and we will refine it and it will become better and better for patients. Just like that's why I always use the airplane analogy. You fly down a little hill, at least you stay in the air, and then you refine this, and then all of a sudden you have a transatlantic flight, and how cool is this? So the key areas that need to work together, where we break through barriers, silos, interfaces, is devices to improve them with local immunosuppression for the islets and enough oxygen. We need better immunotherapy, induction maintenance, ideally with drugs like GLP-1s that can also enhance islet graft survival, that is published, and together with antigenic tolerance. And we need drugs that amplify beta cells, that you can regrow beta cells. So then when the beta cells regrowing, they actually, at least they can do this in mice. In humans, it's always going to come out worse. We already know this, but they can outgrow immune attacks. They run faster than the immune system can run. So there's a hope, at least if you do some of it, the immune system will not be as fast as destroying your islet graft. And with that, we will have eventually a cure. and we will also have the right drugs for prevention. This will go hand in hand. So you asked me to talk for a long time, so you got like a long speech now.
No,
But
it was great. Again, I wanted to sort of lay things out to show the scope of what's going on, and of course the scope of what Michael has to go out there and sell in the sense to get all these different programs funded. I'm going to come back to one of the themes that you touched on it a bit in terms of combinations, and we'll talk a little regulatory stuff. But I'm going to come back to Michael for a minute because I think it's important. Clearly, your funding is amazing at the DRI. You still have a lot going on because clearly this is not, like everything in biotech, we constantly have to keep advocacy, keep awareness. A lot going on, sort of on this front in your shop, Michael. I would love to, and I just pulled some of these sort of off the recent news and the term, certain galas going on in the Hamptons, and you had an interesting run somewhere in upstate New York, I think it was, upstate Jersey, the Wakefern Food Company, a supermarket chain involved. Take a little time to sort of talk about how you cultivate some of these diverse relationships. I know you mentioned the initial 5 families that clearly had a connection here, but talk about some of these and other ones that might not that you're allowed to talk about that may add to this story. I think it's important to show some of the scope of who comes together for funding these types of initiatives.
Absolutely. Happy to. And you put your finger on it. I mean, you mentioned the word sell and sales and in reality, I mean, that's what we're here to do. We're here to sell a product, so to speak, right? And I guess who isn't, right, in their professional lives, but
right.
But the product that we have to sell, you just saw it, you just heard it.
Right.
This is an institute that is on the move. This is an institute that has touched some of the most important discoveries in diabetes to date, and more importantly, it's an institute that is poised to find the cure someday. I have no doubt that one day we'll write the history of the cure of diabetes, and when we do, the institute will figure very prominently in those pages. And so that's our product, and I could not imagine a better product to sell. And we are very grateful to our supporters. You mentioned a number of them. We cannot do it without that support. We cannot do it without the commitment of philanthropists who want to make a difference in the fight against this disease. There are a lot of organizations out there that are focused on diabetes. There's no doubt about that. And there are some very important organizations, and they focus certainly on research. They also focus on education. They focus at times on advocacy and policy issues. And that is all important work. We're a little different in that we focus like a laser on the research, and the research that is curative in its aspirations and in its focus. And that is really, I think, what distinguishes the foundation and our relationship to the donor community. We're giving men and women who have been affected by this disease the opportunity to invest in the cure and to celebrate with us all the victories that we have achieved and will achieve on the road to that cure, victories that will make life a lot easier for the men, women, and children who are navigating this disease. So, I mean, again, what a product. Right. And when we do secure the support of philanthropists, when those philanthropists do support the mission, what we strive to do is to provide them with a stewardship experience that is peerless. In other words, we want to make sure that when you invest in our mission, that you understand the impact of that investment. You see with a high degree of clarity, you have a great line of sight on the impact of that philanthropic investment. And so we connect our donors directly with our researchers. We set up meetings, we set up tours, we provide all sorts of opportunities for our supporters to see and to, I think, gain gratification at what they're seeing, and they see the wisdom, they see the value of their investment in our mission. We strive to do that and to provide that stewardship experience that really is peerless.
Mm-hmm. Mm-hmm. Excellent.
And we are so grateful for that, Michael. I mean, I think, of course, not only me, but all the investigators in the Institute, and of course, ultimately the patients. You make so much of a difference. I think I can only add to that that culture matters. It sets us apart. I'm very keen on that, and probably should say a word on that. I mentioned before the collaborative culture. I also feel what is highly in order these days in science is to re-inject humbleness. And if we stay humble enough, we actually learn faster. And we want to learn faster as a community. This is important for everybody in the world, in the US for sure. And we need to rediscover humbleness. Right? You might sometimes hear this, Ira, but this competition has led to quite some rat race of exaggeration. And I will say one thing, you will say the next thing, Michael might say the next thing. At the end, we would end up in a realm of egregious exaggeration. And then the human mind is such, we fool ourselves. We're thinking, oh, we know all this stuff and we're already here. But in reality, we might be here. And we take these humbling steps back. Right. We learn faster, right? That also means we don't always have to run with the trend. We have to re-overturn stones, sort of saying, is that really so? Really? Right? Especially in an enterprise where notoriously, and we're not going to rewrite everything in 5 minutes, but at least we have to be aware of it. We only publish positive data. You know what this does to science, Ira? We have to be aware that this is the environment we are running in, and it should make us humble. We need to understand the negative data. We have to take a step back. We have to say, does this really work? And then we have to say, can we really fix it? And I think this cultural environment is prevalent at the DRI, at the University of Miami. It has attracted me there because, again, we talked about readjustments. I think science needs this readjustment. And it's very, very important. And ultimately, it will accelerate all of our progress. That's why this is so important. And it will, of course, then if you don't compete and the elbows get retracted, you work together. And if you work together, you can explore interfaces. Between artificial intelligence and modeling beta cell decline, between understanding large data sets in big cohorts like we have in Miami, in making biomarkers, omics, all these things work in the proper way, and really take a very sobering look sometimes at the data that are out there. That's what we need to do. And then we explore interfaces between the immune system, the devices, and beta cell regeneration. We actually have affinity groups that we created in the Institute, and we have an open dialogue that encourages new ideas, new thinking. It doesn't punish anybody. I think that's sort of where we have to equilibrate. It will be, at the end, a tremendous accelerator towards the final goal. Sorry, maybe I spoke out of line, but these are very important for me.
And if I could just add to that real quickly, in my 25, 30 years professional experience prior to joining the Institute, I had the privilege, the pleasure of working with some really extraordinary individuals and at extraordinary organizations, institutions. And I have to say that I have never seen the degree of collaboration anywhere else than what I'm seeing at the Institute. It is extraordinary, the degree to which the clinician scientists, the researchers are collaborating, are integrating, basic translational and clinical research in ways that are accelerating discoveries. I've never seen anything like it, and I think that's a very big reason why the Institute has touched so many of those discoveries it's because of that extraordinary collaboration that takes place under the roof of the Institute.
Mm-hmm. Mm-hmm. Well, yeah.
I mean, go
please.
ahead. Sorry.
No, I was just, I'm glad you both brought up, collaboration, but Matthias, you were mentioning interfaces, and I thought this would be an interesting time to head in a related direction for both of you. And Mike, I'll come back to you with your version of this question, but I thought, Matthias, as you were talking before about things like the GLP-1 work and the sort of remodeling the islets. I looked back at some of your papers. There was a really cool one from back in 2008 in terms of how one eliminates persistent viral infections, looking at PD-1 and IL-10 and these interesting combinations of approaches. It was the combination approach ultimately that took something like HIV, which was a death sentence at the end of the 1980s, to a fairly livable condition. Obviously, the PEPFAR program, and we profiled Ambassador Nkangosong in $200 billion, but nonetheless, with the right money there, the strategy of combinations, and Gilead, some of those other shops got really good at this. Are there interesting things that you have learned or potentially can learn from sort of that model? Because clearly, yes, we can develop each of these interventions as you're talking about individually, but it's really putting them together that's what eliminated HIV. I think, and I could be wrong, but the proper combinations, and I don't know if this requires proper regulatory structure, what have you, getting them combined early on, getting sort of these combination interventions together, I think is one of the holy grails to what you're doing as well. And I just love your perspective on that. If there's things you can learn from the HIV industry, from other parts of the pharmaceutical industry, please go in any direction you want with that one.
Look, I couldn't agree with you more that combination therapies are the way to go. I said it already before here, and we wrote lots of papers on there. And it's mechanistically founded because we got to do something for the beta cell to recamouflage it, and we also got to do something about the immune system, especially in a selective way. I can give you some of the learnings that are quite interesting in my view from a published paper of probably the largest recent onset type 1 diabetes adult trial ever run in 300 something patients in Lancet Endocrinology. We published it. So we combined the GLP-1, and this was a Novo Nordisk trial, with a blocker of the immune system systemically at that stage, anti-interleukin 21, based on good animal data. And you alluded to regulators. Yes, there has to be something there before you do this. At least you need to be clear that this can be an additive effect, not necessarily synergistic, but an additive effect. And the key with these combinations is that you say, well, both of the drugs, any drug has side effects. You hope, of course, you pick ones with very low degrees of side effects for these combis. But the side effects should occur in different areas, meaning in different sectors, right? But they should synergize in their efficacy in protecting the beta cells from immune destruction. So from this trial, we learned it reached endpoint. It was successful. It preserved C-peptide, reduced insulin dose. These were pre-specified endpoints for this trial. We could show that there's at least an additive effect. We also learned we need to give the drugs to have the effect. When you withdraw the drugs, it doesn't reset anything, which you can look at this in two ways. You can say, well, that's bad. We want to reset the system. The other thing you can say, well, good. If you take the drugs off, you also don't have the effects of the drugs anymore. So you retain the control, like you take and it's actually normal. You take an antihypertensive pill, you might have some side effects, but you control your blood pressure. By the time you stop, you don't control your blood pressure, but you also don't have the side effects anymore. So that's how it usually works with anything, with the statin or any other drugs, right? The other thing we learned, which was quite interesting, this was a post hoc analysis, is that it seemed that the GLP-1 component in patients who entered the trial with a higher degree of beta cell function, as evidenced by C-peptide, right? Mm-hmm. That GLP-1 component was the sole driver of the function preservation in these patients. So that means as long as you have enough beta cells, at least in these trials, you did not need the immune modulation. This led us to propose, this is of course just a hypothesis, it would have to be proven, hopefully at some point in the prevention trial, that there is a stage in the disease, and that goes along with the pathology of the disease, early on, stage 1, when you have autoantibodies, where there's very little cells that you find attacking islets, if any. It's very spurious. At that time, it seems, before all hell breaks loose, so to speak, and the immune system gets activated. So this is not a linear process. This goes like this, and then boink, you fall off a cliff. So before the immune system gets activated, you can insinuate that protecting the beta cells from recognition is the main objective. And I don't want to say that GLP-1 drugs is the only way to do it. There have been interesting study with verapamil that needs to be repeated.
I saw that one.
And there need to be tolerable drugs by definition. If you get, or I get diagnosed tomorrow, I have 2 autoantibodies, you're not diabetic, the willingness from a patient perspective to take anything major will be low. You want to have a very tolerable drug. So that we have to choose. And then it also tells us in a way that the immune modulator precisely then important when you undergo major loss of function of beta cells. And that is where TZIEL, the anti-CD3, is licensed right now in stage 2. This is where probably in our combi trial, the anti-IL-21 would have the best efficacy, because when you lose beta cell function, this is, in my opinion, pathologically the time when the immune system has realized it, wrongly so, it should kill the beta cells, and you have a lot of immune activity. And that's sort of your flair, so to speak, when you know how to treat the disease with an immune modulator. That concept of combination, that you start with well-tolerable drugs, ideally in our mind with antigenic tolerance that selectively takes out the cells that attack the islets. This was the one trial I was referring to early on. That combination should be done. And then when immune attack ensues, you can use systemic immune modulators. By the time you lost all beta cells, you stop these immune modulators. And islet transplantation is the other way around. You use the immune modulators when the islet comes in, and then you use the milder therapy as a maintenance therapy. Regulatory-wise, I think that is more complex. We will need probably help to together understand what it means to have these combination trials, because other than in oncology or also HIV, a type 1 prevention trial going to take 5 years. We don't have so many shots on target. This is not a it's not like a recently diagnosed stage 4 pancreatic cancer trial where you can have a short time to endpoint. These are complicated trials. So as a community, meaning all of us in this world, we have to, if it were my way, we have to favor these combinations. We have to be prone to try them, maybe in an adaptive design, and we should do more of them, but we also should be cautious not to shoot all our bullets with one drug trials. This is not ideal because the stuff takes so long. And there's no easy way around. You need enough individuals, you need to wait long enough, and it's a heterogeneous disease. And because of that, you need to run a large enough trial with a long enough duration. And if you don't do that, your RCT is going to give you lousy data. If you have lousy data, you, depending how humble you are, you're in the realm of wishful thinking, or you have nothing. You know, and I'm a strong believer that we have to have large enough trials. We got to do this right. We got to learn from these trials well, and we can have hopefully adaptive design. Maybe we can have at some point historical or jointed control groups to alleviate the burden enrolling too many patients. Maybe we can even at some point, if we have enough data collected, that's of course then the elephant in the room, virtual control groups, but I don't think we're quite there at this point, certainly not for prevention. But that's where these things should be going in terms of a trial design, right?
Yeah. No, it's all extremely
but combinations,
excellent points.
Yes, combinations, we will need them just because we have different players at work, right?
I'm excited that you're thinking about all these things, though, and again, you know, I appreciate you taking the time to lay that out, because I sort of want to show the sort of the multifaceted nature of not just what you're doing, but the way you think about you know, the early discovery all the way to how ultimately these gets to patients. So I think that's extremely important. And I'm glad you walked us through this. Michael, a related question, and bear with me on this one if you would, but I'm sitting here in downtown Philadelphia. I know back in, I think around 2012, you took on the chief development officer role down the street here at Fox Chase. I'm a Philly kid that, grew up on the Philly pharma scene, and one of my heroes that we lost recently, none other than Dr. Beatrice Mintz, used to work over there for many years. And a lot of the work that B did in terms of animal chimeras and transgenics and immunotolerance and cell differentiation back in the 70s all make for wonderful tools today. What with Matthias and everything's going on at DRI. I just got to ask, did you have any interaction with B when you were at Fox Chase? And are there any interesting things to leverage in terms of, as we're talking about sort of all the intersections of these diseases, from the cancer folks or the Alzheimer's folks, or whatever the case may be, because we're finding out so much about whether it's the models or whether it's the disease processes that potentially we could synergize across these different types of funders that look at these types of programs.
It's a great question, and just for the record, I'm a Philly kid too. Born in West Philly, grew up in Chester County. So I did have the pleasure of meeting Dr. Mintz. Fox Chase is just such a wonderful story. It's arguably the first, I think Memorial Sloan Kettering may take issue with this, but arguably the first comprehensive cancer center, NCI designated comprehensive cancer center.
Yep.
More than one Nobel laureate kicking around those hallways. Just their contribution to the field, has been extraordinary. When I was in discussions for this role, Matthias and I had a series of conversations, and we talked a lot. And of course, Matthias has a background in oncology as well. We talked a lot about the relevance of oncology, the relevance of cancer research to the work of the Institute. And there's a lot of crossover there, no doubt about it. I think Matthias is clearly in a much better position to kind of talk about the scientific relevance there, the overlap. I would just say at a very high level, again, I think the basis of our progress with cancer, both understanding it and treating it better, so much of that progress really comes out of our nuanced understanding of the intersection of cancer and the immune system. Mm-hmm. These game-changing therapeutic strategies in immunotherapy, T-cell transplantation, CAR T transplantation, engineered T cells, and checkpoint inhibitors have transformed the treatment of many cancers. And again, I think that we're going to see that kind of progress in diabetes research and care based on our more nuanced, better understanding of the interaction between this disease and the immune system. I want to focus a little bit on a different kind of overlap in this space, and one that kind of is more relevant to what I do day in and day out, and that is the collaboration between like-minded organizations in this space and also in cancer. I guess that was a lesson I really learned in cancer, is that when two or more organizations that have skin in the game, that have a shared mission, that have a shared sense of what we need to do to get to where we want to go, when you see that kind of collaboration, that's where you really see progress. I don't think there's any doubt that, at least in my mind, that no one organization is going to deliver the answer. I think that when like-minded organizations come together and pull in the same direction, that's when you're going to see real progress toward the cure. And I have spent a lot of my time professionally cultivating those kinds of collaborations between organizations that I either lead or have been involved in, and other organizations that, again, have a very similar mission and are focused similarly to the way we go at the work. And it's really truly a case of 1 + 1 = 3. And so there are a number of examples I can share. It's just historically, we've had a strategic alliance with the North American building trades unions that has lost it for decades now. NAB2 has been extraordinarily generous. This is a disease that affects a lot of the membership of that organization. They have been exceedingly generous to the Institute over the years, have supported us, over $80 million over the years, including building the building that the Institute calls home in Miami. And they continue to this day to support us on an annual basis in a very significant way. So that's a great example of a historic, but also current meaningful relationship between the Institute, the foundation, and an external player who is making a difference. Another example, when we're in a partnership with Instacart Health. This is an offshoot of Instacart, which is, obviously a paradigm shifting company that is rewriting how people get their food, obviously. And so we're about to announce a collaboration with Instacart Health, which is a division of Instacart focused on providing the public with actionable information about healthful eating specific to disease states, so that if you're managing, a cancer diagnosis, these are certain lifestyle choices, certain nutritional strategies to address that. And obviously with diabetes, where nutrition and diet are, critical to successful management. We are going to be in a position now, and we're very grateful for this opportunity, to engage the Instacart Health constituency, their customer base, around healthful eating. We're providing all sorts of resources and information to that end. Again, formally, more later on this. We will be announcing it shortly, but we're very, very pleased to be in partnership with Instacart Health. And just to kind of round off this thought, one of the first things I did when I came on board was to sit down and meet with my counterparts at some of the high impact organizations in this space, Breakthrough T1D, formerly known as JDRF, of course, ADA, other significant players in the space, all with the goal and the focus on how can we come together, pull in the same direction, pull resources, and get at this with more efficiency and catalyze through that partnership real advancements in the field. So this is front and center in terms of my focus as the chief executive of the foundation, and I'm really eager to establish and sustain these partnerships.
No, those are all awesome stories. I'm sitting here smirking as you're telling me that, just because one, my mother-in-law is it'll be great she can learn because she uses Instacart all the time. But at the same time, I was just coincidentally last week checking out the Food is Medicine conference in Chicago, and there was someone from Instacart Health there. I was like, I didn't even know this existed. So that's very timely, and I appreciate, Mike, that you bringing that up. I realized time flew. It was an amazing discussion with both of you, but while we have you at the top of the hour now, final messages. I'm sure we could talk for another couple hours because it's just so also interesting. But final messages about other public-facing stuff that's happening at the organization. Michael, you in terms of some upcoming fundraising initiatives. Matthias, anything else on the R&D horizon that we didn't touch on that you can mention as we enter 2025, or anything else you want to talk about while we have you? Please, both of you take the floor.
Matthias, you want to jump in first?
Sure, I can. You alluded to cancer and collaboration between different funders and organizations. I mean, what I'm particularly excited about is we have now these organoids for cancer as well as islets. We can put them under immune attack. I think we can now learn much faster. We have collaborated with a company in Sphero in Switzerland on this. They have now banks of cancers. We can make different immune-evasive islets. And you know what we need to learn from this, what I'm excited about, We need to get a roadmap of which molecules matter most, either for attacking cancer or for keeping islets protected. And the cross learning is there. You know, the checkpoint inhibitors. There's the ones that interfere with the PD-1 ligand pathway are the only ones that induce diabetes. So there's a message there for us for type 1 diabetes. That's what I mean with cross learning. And why do we need a priority map? Because, as we alluded before, the trials in prevention and trials in islet transplantation, they're long, they're complicated trials. So we need to get our ducks in the row before we embark on these clinical investigations, so we deliver the right shots on targets. So that's science I'm super excited about that the future is bringing. In terms of a general concluding message, I stay with my message of collaboration and humbleness. It will serve us well. It will make us all faster, not only, in type 1 diabetes and cancer. It will probably make us faster when we look, at the COVID pandemic. We look humbly. We take the learnings. What did we do well? What didn't we do so well? It will make us faster and better as a scientist and as humankind in progressing with science on the right path. Anyways, to you, Micah.
Awesome message.
Thank you. And again, the product is phenomenal. I just, want to just be really clear that I am so impressed with what's happening at the Institute under Matthias's leadership. It is an extraordinary enterprise, and it's a privilege to be associated with it. I would just maybe end with almost a naked appeal here, a somewhat self-serving appeal that I just think that anyone who wants to make a difference in the fight against diabetes, who wants to invest in the cure, to look at our organization, because I think from soup to nuts, we can demonstrate that your philanthropic investment in our mission will have an enormous impact, and that we can one day celebrate together when we do defeat this disease, and we will. And when we do defeat that disease, we will celebrate together with our philanthropic investors, with our donors, with the clinician scientists and the researchers and the policymakers. But if anyone wants to directly impact the trajectory of research, our understanding of this disease, and that how we translate that into new therapies and into one day the cure, look no further. Diabetesresearch.org is our website, and I would encourage anyone who wants to learn more about our organization to take a look.
And we will put that link in the bio of the show. Really awesome messages, and really wishing both of you the best. Mike, you on the fundraising front, Matthias leading this amazing set of research programs. Again, for everybody that is going to be listening to this episode of the show across the various podcast networks, or who'll be watching on our YouTube channel. Again, you've been spending time with Michael Burton, Chief Executive Officer of the Diabetes Research Institute Foundation, and Professor Dr. Matthias van Herrath, Scientific Director, Diabetes Research Institute, and Stacy Joy Goodman Chair at the University of Miami School of Medicine. Michael, Matthias, again, I want to thank you for both taking an exceptional amount of time out to talk about what you're up to. Thank you for everything you do for this particular significant unmet medical need. And as we like to say on our show, thanks for helping to create a better tomorrow for millions of people out there. Awesome story.
Thank you, Ira.
Thank you, Ira. Thank you
Great
so
having
much.
you.